So when we're going to be talking about diffusion and osmosis in this section, we're talking about the movement of molecules, okay? And so what the question is, well, what is a molecule? And so a molecule is the building block of matter. All material is made of molecules, of these little particles, things like glucose, carbon dioxide, oxygen, water. These are molecules, building blocks of matter. And when we talk about osmosis and diffusion, we're talking about how these molecules move, okay? How they can be transferred from one place to another. And we're going to talk about two specific cases, osmosis and diffusion. So diffusion is when molecules move from an area of higher concentration to an area of lower concentration. High to low concentration. That's the key thing for you to remember. You could sort of think about it this way. Molecules don't like to be crowded together. So the same molecules, they don't like to be in one crowded little area. Okay? They will naturally, on their own, tend to spread out. To go, and we call where they're all close together an area of high concentration. To spread out is an area of low concentration. You can imagine it this way. If I were to take every 7th grader in our school and I were to bring them into this room and I close these doors. Okay. Every 7th grader standing could probably cram into this room. Would it be very fun? No. Would it be smelly? Probably. But we could probably you could all probably fit standing real crowded. That would be an area of high concentration. If I did that, and then after 5 minutes I open up the door back here, what do you think the 7th graders would do? Everyone would move over there?
No,
Yeah, about how many?
like half.
Probably half. And then everyone would have a little bit more space.
Yes.
If I open up the door to Miss Bean's room next door, what would happen?
A quarter.
Yeah, probably another section would spread out to give themselves some space. Now, I wouldn't have to probably say, hey, half of you go over there in Mr. Arcuri's room. You would just sort of do it on your own. You wouldn't take any energy for that to happen. Well, that's like diffusion. Molecules don't want to be tightly packed. They tend to spread out. So, for example, let's talk about a couple examples of this. Every morning when Mr. Arcuri comes to school, he comes in that door and he puts his perfume on in my classroom. And he puts it on way in that corner, and just 10 or 15 seconds later, when I come in the room, I could smell it all the way over here. How can I smell that perfume from that corner of the room to here? Nick?
Because we kept the door closed and the smell must have got around the room.
Yeah, how does the smell get around the room? Yeah, when he sprays the perfume over there, the perfume molecules are all in a little cluster. They're in a concentrated area. And they just tend to generally move apart. And they're spread out all across the room, and that's why I can smell it when I come in the room. That's an example. If I put a drop of food coloring in this water, the drop of food coloring is a high concentration of these molecules. What's going to happen to it? It starts off under a high concentration, but what are these molecules doing? They're spreading out. The molecules are diffusing throughout the water. And after a little while, we'll see that that's going to look just uniformly red. The molecules have diffused. So those are some examples of diffusion. So,
Am I allowed to tell Mr. Arcuri he smells good today?
sure, go ahead. That'd be a nice compliment. Now, osmosis, so molecules diffuse just like through water, but they also can diffuse through a cell membrane. If a molecule is small enough that it can fit through the cell membrane, it will diffuse. It will go from higher concentration to lower concentration, okay? As long as it can fit through. Things like oxygen, carbon dioxide, water, are small molecules. They can diffuse through the cell membrane right into the cell. A special type of diffusion is called osmosis. Osmosis is when it's water that is moving. When water goes from a higher to a lower concentration through a membrane, we call that osmosis. So let's look at an example. If I had this U-shaped tube, okay, and it's two parts, you could separate it. And in between the two parts, I put a semi-permeable membrane, like a piece of this dialysis tubing. Okay? And I split it up. And then on this side of the dialysis tubing, I just put plain water. So let me do this. Yeah. So let's say on this side, I put something that is 2% salt. What percent water is it? 98% water. Okay, here's the membrane separating them. Then on the other side, I put a solution, let's say that's 15% salt. What percent water?
88.
Okay. Let's think about this. Now, where is there a higher concentration of salt? Where is there a higher concentration of salt? Lily?
The one to the right?
Yeah, this one. There's 15% salt. Here there's 2% salt. This is a higher so which way would the salt want to go based on diffusion? Sophia?
To the left.
Yeah, to the left. We want to go this way. But let's say this membrane is not permeable to salt. Well, it can't go anywhere. Salt can't go from right to left or left to right. So the salt stays where it is. Where is there a higher concentration of water in this example? Bailey?
The side that has 2% salt.
Yeah, this is 98% water. What's this? 85. There's a higher concentration of water here. Which way does the water going to go? Based on osmosis.
The right.
To the right. High concentration to low concentration. And if this membrane is permeable, water can get through it. Water is going to continue to go on this side. And it's going to move over there. And what this ends up looking like, so if water is going this way, what's going to happen to this water level? It's going to go up. What's going to happen to this water level?
Stay down.
It's going to go down. And that actually will happen.
Oh.
Yes.
You do know that the molasses is dripping on the table?
Oh, is that? Yeah, that's all right.
Over here.
Okay.
Molasses jar.
Rotate this though, let's see. All right.
What's happening?
Well, we'll look at that in a minute. You'll see. You'll see it obviously. Okay. So that's osmosis. Now this is important in ourselves as well, because our bodies. So let's say our blood, for example. Our cells are mostly water. The cytoplasm of the cell, most of it's water, 90-something%. Okay. But if we take our cells and we place them in very salty water, or if we place them in just pure 100% water, well, things happen. If you look at this example, okay, these are cells that are placed in just a solution that's balanced. So let's say our blood cells are 96% water. If I put them in a container that's also 96% water, what do you think happens? If the concentrations are equal, what do you think happens? So,
Well, if it was like water, then if you put something in, sometimes it rises, the water
okay.
rises.
But I'm saying if the blood cell and the surrounding liquid is equal in concentration, is water going to go into it?
No.
Is water going to come out of it? No, they're balanced. It's going to stay the same. And that's what happens. When you go to the hospital and they give you IV fluids. Like sometimes if somebody's dehydrated, like somebody ran for them and they got dehydrated and passed out, they'll give them an IV, they'll give them a needle, it goes right into one of their veins, and they have a liquid that's going into them to give them more hydration. But they do not give them just water in that bag. Okay? They give them what we call a saline solution, something that's balanced with their blood cells, so that their blood cells don't take in water. Okay?
So like, if it was like, say you did like 100% diet, 100% water,
I don't know what you mean. It can't, because if you mix two things, they add up to 100%. Yeah.
Well, yeah, like if it was 50-50.
Then there would be no, it wouldn't go either
So
way.
would
It'd be
you-
balanced. So, cell A is a cell that has an equal concentration of water. Cell B is what would happen if we placed cells in a low concentration of water. If we had a very, very salty solution, let's say it's 20% salt, 80% water. Our blood cells are 96% water. So because of osmosis, water would go from our blood cells into that very salty water, and they shrivel up. They lose water from their cytoplasm and they shrivel up. If we take cells and we put them in a solution that's very high in water, distilled water, that's just 100% water, now there's a higher concentration of water outside, a lower concentration of water in the cell, and water goes into them, they swell up, they could even burst. That's why when you get IV fluids, they don't just give you pure water. If they did, it would be dangerous, because then that pure water, because of osmosis, would go into your red blood cells, they'd swell up and possibly burst.
Oh, God.
Sophia?
This is a little like on topic but off topic.
Okay.
Do they consider our cells as like animal cells?
Yes. Yep. All right. So let's pause here for now with our notes.
Okay. Are we gonna do notes at all?